So we have 100 billion nerve cells, and we have 100 trillion points of connections or synapses. And as you heard, autism is a disease of the synapse, but also can be a disease of our DNA. So how can we fix it? So about 20 years ago, we started looking to see how the brain protects itself. And we looked at glial cells, how they protect the nerve cells. And we discovered a new protein to science that we called activity-dependent neuroprotective protein, because it protects the brain against electrical blockade, which is what happens in neurodevelopmental diseases and also in neurodegenerative diseases. So we discovered activity-dependent neuroprotective protein, in short, Adnp. And now there is an Adnp syndrome, and we predicted it quite a number of years ago, actually at about the same time. And what we also did, and I'll tell you about it a bit later, we also derived a very small fragment of an Adnp that can enhance Adnp activity. So a loss of Adnp, we wanted to know what it does in the whole animal. We knocked it out and we found that it's a no-brainer. There's no brain. So knocking out Adnp, you don't get an animal model, you just don't get a brain. Adnp is essential for neural tube closure. However, if you look at the haploinsufficient mice, so these are mice that have only one copy of the Adnp gene, they are alive, but they suffer from cognitive and social impairments. So this actually led to the discovery of the Adnp syndrome. And here you see 8 children with the Adnp syndrome. They have very distinct facial similarities. They have a very thin lip, a high brow, and a distance between the eyebrows. And they all share intellectual disability. They have severe speech delay. They have a severe, some of them, a severe motor development delay. They have sensory processing disorder, sometimes insensitivity to pain, and some have also sleep disorders. And again, looking at the Adnp gene, which we cloned so many years ago, you can see some of the mutations here. And you can see that it's a very busy gene. It's a very large protein. The protein out of the gene is very large. And it has many zinc finger domains and a homobox domain. And also it has interaction domains for other proteins that have been implicated in autism. For example, proteins that regulate translation and other proteins as well. And it is specific for vertebrates. So it is specific for animals with a higher brain structure. It's very highly conserved. So one of our first findings was that it interacts with a complex, a chromatin remodeling complex that's called the SWI-SNF complex. And this complex also interacts with the MECP2, which is the Rett syndrome gene, the FMR1, which is the Fragile X syndrome gene, and also with the gene called disrupted in schizophrenia. And as you saw, Adnp, we also found it regulated in schizophrenia. So when we look to see what kind of genes Adnp regulates, how come it is so important for brain development? So during embryonic development, it regulates 400 genes, more than 400 genes, that are essential for neurogenesis, organogenesis, regulation of transcription, and also lift transport, little vacuole and coagulation. One of the genes that Adnp regulates is apolipoprotein E, which is the major risk gene for Alzheimer's disease, so associated with cognition. Another gene that I will talk about a little is BMP1, which is bone morphogenesis protein, which actually affects both brain development and also, of course, bone development. And why am I saying that? Because recently, looking at 55 or 54 Adnp children, we found that 44 of them have very early tooth eruption. And this is associated with Adnp regulation of bone morphogenesis. So this is some of our looking at RNA set from the human lymphoblastoid cells and also comparing them to the mouse model that we have. And Adnp regulates genes that are associated with brain morphogenesis, of course, and also with bone development. And here you see the original finding actually here was made by a mother who is Sandra Bedrosian's sermon, who leads the Adnp Kids Research Foundation. And you see here her two twins. So this is the normal twin at the year of 1, and he almost doesn't have any teeth. And his brother, and this one is Adnp a child, and you see here that he has at the age of 1 a full mouth of teeth. And these are other children with the Adnp syndrome, and also at 1 year of age they have a full mouth of teeth. And they also, the Adnp child lost his teeth a year earlier than his twin brother, and the tooth also had a lower animal thickness. So the teeth develop earlier, and they have a lower animal. So this is actually a way to very early diagnosis of Adnp children. So this is something I actually would like to know, whether in Israel, among the 17,000 or something like that, autistic children that are children that had very early tooth eruption, and then we can look at them and see if they have the Adnp syndrome. So this is very interesting to me. And you will see why in a minute. And also, when we look at the mouse model, so mice don't have primary teeth. They only have permanent teeth. So in the mice, they also use dysregulation, but the teeth come out actually late, the permanent teeth. And you can see it here, so the tooth bud and the dental sac, and you see it here by micro CT. And also you see here that the area in the mouth of where the teeth are supposed to be growing is actually innervated with Adnp nerves. So we looked both at by RNA-seq, we looked both at lymphoblastoid cells derived from patients, and we looked also at our mouse models, and we found that Adnp regulates ossification, skeletal system development, Wnt signaling, limb morphogenesis, embryonic skeletal system morphogenesis, and osteoblasts. So it's not only the brain, it's also the bone. And here is just a comparison between, again, genes that regulate the BMPs, that regulates the bone and the brain. And you see that if there is a mutation, this is the different lymphoblastoids with different Adnp mutations. In all of them, we see dysregulation of the BMPs. And so we also calculated what is the prevalence of this syndrome, and we estimate that there are about 6,200 children with Adnp mutations in the developed world. And now we have this marker, and maybe we will get to know many of them much quicker. Right now there are about 155 children that have been sequenced and shown to have a de novo Adnp mutation. So it's all de novo mutation. It's not inherited, and it's a heterozygous, so it's a dominant mutation, and it's the only difference between the child and the parents are not autistic. So it's a de novo mutation that drives the autism, the Adnp syndrome. And here are the locations where the disease is found. We have right now only one child in Israel. And Joe already, Joseph, sorry, Dr. Buxman, already alluded to that Adnp is one of the leading genes causing autism syndrome. And you see here one paper that just came up recently. You see that Adnp is here next to SINGAP, etc. We don't have shunt here, I don't know why, but there are about 10 something like that genes that are leading, and Adnp is among them. And here you see some more Adnp genes. And there is a very good website from the SPAR or Simon Foundation where you can find out there are many case reports about Adnp, and we just now have a paper that will come out, I hope, soon. I've been promising this for a while, about 78 children. And Frank Kui from Antwerp is leading this. And so in terms of case studies, we wrote a case study recently about one child with a mutation in the protein in tyrosine 719, which actually breaks the protein into half. I should say that all these de novo mutations are either truncating completely, so you have a shorter protein, and both alleles are expressed, or it is a French shift, and then again you have a shorter protein. So here you have the case study of this child. P1719 is the most prevalent mutation in Adnp. It's like 20% of the cases. And you see, one of the major things that you see in these children is a motor delay. So they are all they're intellectually disabled, and they have also a severe motor delay to the degree that this child started walking when she was 3.5, and the parents were told that she will never walk. And you see here some more children, and you see that they have walking aids, but eventually they do walk. And when you look by MRI, there is a developmental delay of the brain development, and here the hippocampus is delayed in terms it's more rounded, vertical and medial position, than normal. So there is a difference in the, also in MRI. It's not a very marked difference, but the brain develops probably slower, and the differences are noticed by MRI. So Adnp, as I said, is interacting with the SWISNF chromatin remodeling complex and regulates directly quite a number of genes, and we found that it can act as a transcription factor, but it can also be found in the cytoplasm, where it interacts with the microtubules, which you heard about the actin, so this is again the cytoskeleton, and the skeleton of the nerve cell is essential for the structure of the nerve cell. And if you have less Adnp when the mice age, actually they develop something that looks like Alzheimer's pathology with neurofibrillary tangles, and this is a protein called Tau, like Tel Aviv University, and it's a microtubule-associated protein that actually binds to the microtubule structure and also to the microfilament structure which contains actin, where Schenck is interacting with. So I hope I didn't mix you up completely. So if you look at the mature animal, or in the developing animal, Adnp regulates thousands of genes. So one of the beliefs is maybe that these genes that are associated with autism are key regulators of gene expression and of genes that are important for the brain. So one of the genes that Adnp regulates is tubulin, or the subunit of microtubules. It also regulates channels that are important for ion movement in the brain, and also transporters that are important for neurotransmission in the brain. And here you see the many differences that we see with age and with sex. There is a differential sex. Okay, so I have only 5 minutes, so I'll go very fast. Summarize the 20 years of work. So this is we took out a very small fragment of Adnp, we call it NAP, and this is the drug target. We know how it works because we found that it interacts with the microtubule end-binding protein, and you see here it has a motif that binds to the microtubule end-binding protein. It regulates synapse formation. You see it here in green. So you see when you add NAP, you have many more connections. And it regulates the microtubules. So these are the microtubules. Without the microtubule, you won't have a functioning brain. So you need to have these microtubules to build axons, which are very long. And you have the microtubule end-binding protein, which contribute to the dynamics. And so I'll show you the result because I do not have time. And the result, I don't know if you can see it, but it's a movie here. And you can see that when you add NAP, the small fragment from Adnp that binds to the end of the microtubule, you actually increase microtubule dynamics. So you increase the movement of material in your brain cells. And and if you don't have Adnp, this is going to be slower. And this is why the brain of the autistic Adnp child is a bit slower. And also, it requires this protein Tau for activity. If you don't have Tau in the cell will die when you had Adnp or NAP. And here you see it when you had NAP. The only cell that survived is the one that has Tau and NAP. And our working hypothesis here in terms of the activity is that, and this work was just published in Molecular Psychiatry, so it's 2017, so you can read it because I don't have time to describe it in depth. But what I wanted to say is that actually the microtubule end-binding protein bound to the protein Tau, And tau deposition in Alzheimer's disease has been directly associated with decreasing cognition. So, and of course, microtubule function is associated with all this, as it is a disease of the synapse, it is associated with many proteins or many genes that are associated with autism that are found in the synapse. And what we show here is that the addition of this small fragment of Adnp NAP enhances microtubule end-binding protein to tau and tau binding to the microtubule shaft, which will stabilize the microtubule. And it enhances it by about 20-fold. So we know exactly the mechanism, and it does it through also interaction with Adnp. And of course, when we have, just going from that, when we have reduction in Adnp, we will have reduction in axonal transport, also in the intact animal, which we measure in intact animals. And you see here, measuring by giving the animal manganese, you see the manganese move in the olfactory valve. And you see here that the signal intensity of the manganese, this is in the wild type, and this is when we look at the Adnp haploinsufficient animals, and if we treat them with a fragment from NAP actually, 4 amino acids that actually binds to the end-binding protein, we enhance the axonal transport. And enhancing axonal transport will enhance cognition. And this is different between males and females, as you can see. And getting back to the protein, to the peptide NAP, it also enhances cognition in the animal model, and this is a Morris-Watt maze, and you see, and they have to remember how to find a new platform, and if you give them, if you look at the animal with Adnp haploinsufficient, it doesn't learn at all, doesn't remember where the platform is, you give it intranasally NAP, it remembers. And I'm not going to show you all this, but also I'll show you this. In a clinical trial in amnestic mild cognitive impairment patients, you give them NAP intranasally, and it was called the Vunetide also. It actually enhances cognition. So this is short-term memory. It also enhances activity of daily living in schizophrenia patients, as you can see here, with a very significant p-value. And this is like the ability to write a check, to travel with the buses, etc., with public transportation. And again, now, Cornish Neurosciences, a company based here in Israel, is going to do a clinical trial. This is the clinical trial design. I will talk about it next year because they asked me to finish. So thank you very much. Sorry for the very much.